Okay. Are again, it's oncogenes, and the other one that that I think is worth noting is the tumor suppressor genes, and it's really fascinating. If you're all capable of reading this now, there's a wonderful book on cancer that won a Pulitzer Prize, and it's called the Emperor of All Maladies. And it must be about 9 years old now, and this he basically wrote about cancer almost biographically, and it's brilliant. I think his last name is Chatterjee. Uh, I think he's an oncologist, and it's really an interesting read about how long people have been suffering from different kinds of cancers and the approaches. And really, his analogy to this are the accelerator and the brakes. So normal an oncogene, basically we have what are called proto-oncogenes. They're always they're in the off position. Basically, it's almost like it retards your accelerator, if you want to think of it that way. And what happens when a virus damages that as it gets incorporated with the viral genetic material, it can switch that from the off to the on, read that as oncogene or tumors. And so basically it's accelerating the cell cycle. Think of it that way. It's accelerating reproduction in a variety of ways, including the cell cycle. The other are tumor suppressor genes. Those are the brakes. Your brakes normally work. But what happens is the genetic impact of that viral infection can damage those brakes and you lose them. Now imagine if both happen simultaneously. You'd be accelerating and you have no brakes. I mean, you're going to get into a crash. That's maybe the little nugget that I think that I did want to share with you. That I think I covered everything else in that viral chapter PowerPoint that we use in micro so much. So let me do the genetic one, which you've all had 16 times, but I do it mostly for the conjugation, transformation, why isn't this Xing out? I give up. I demand a recount. Love that. Another one, the transposons, something I would remember, and because they can be moved by themselves, typically with the aid of plasmids. They move in and out, and you can reproduce sort of irregular patterns of identical genetic structure. And she studied in what I used to call Indian corn, but of course we now call Native American corn, because we're politically correct. I've done that routine for you. We don't have Eskimos, we have cold Americans. Right? See, a double entendre remark, I would say that intuitively. Intuit.
Oh. You're on a roll this morning.
Yeah.
Like, honestly.
Actually, it's Shibata. I don't know. I'm one of those highly irreverent. I thought we were going to, my wife, I saw her for the first time for half an hour last night. And she came home, and then she almost had to leave again because we have that blink system to monitor what's going on with her mom. And the system crashed. And I'll take the dog out, I'll run down there. Maybe there's something wrong with the internet. Unfortunately, I got back in and must have just winked out because it's back on. Not easy with these older relatives. But she studied the corn and looked at the patterns, Nobel Prize worthy, etc. So all of these, the resistance nugget is so terribly important. This is the idea of mutations, and really, if you want to think of it from a microbiology perspective, this is we have evolved, and you guys didn't have me for micro. And it's a big talking point. We have evolved Staph aureus, not once but twice, in the span of 80 years, roughly since the implementation of treatment with penicillin, which is around 1941, when Pfizer Labs dedicated everything to making penicillin for the effort for World War II. You guys don't know I'm a World War II buff a lot. And first, 90% were susceptible to penicillin. Now, by 1970, 90% were not susceptible. So we started using methicillins, and now Look at that number. 2004, 60% strains from hospitalized patients were methicillin resistant. 2.3 million. This is not necessarily new data. This is probably 6-year-old data. It's scary. That's the genetic fragility of bacteria that's so important. And then you get into these whole things with regard to this isn't that terribly relevant, and that's not relevant. And I'm not going to go over definitions, but they might use those on the Mcat, so you understand what auxotroph versus prototroph is. So the auxotroph does require, and that's part of it there. And of course the nomenclature, because again it's like in plants, because they used to think these were plants because they had a cell wall. Bacteria. So that's where you hear the term normal flora. Now we use microbes is more scientifically correct. But there you either have the wild type, the typical type that you find, or it's a mutant. So that's the terminology that's typically used. With the idea, what are the things, there are different things that make these organisms mutate more rapidly. We have three major things from eukaryotic cells versus prokaryotic cells. Eukaryotic cells, double-stranded DNA, dominant and recessive. And so inherently, if it occurs on a strand that's not coded, if it's heterozygous, then if it's on the recessive strand, it doesn't demonstrate. So, tree, forest, nobody hears it. That deal. If it's double dominant or double recessive, it has to be on the strand that it basically is being demonstrated or being utilized. So at least you have a 50-50 possibility. That's one. Two, messenger RNA processing. The introns and the exons. So if it is on an intron, on an insertion sequence, that's excised. Only the exons are coding for proteins. Doesn't matter. Lastly, DNA polymerase 3, a much more sophisticated proofreading than bacteria, which have something called restriction modification enzymes. So they are much more likely to be able to not only catch the mutation, but repair the mutated side. Restriction modification basically is mismatch repair. It's a 50-50 proposition. You probably did that experiment with the E. coli and tagging it in the fluorescent light and how many colonies. There's a kit from Edvitech that we use all the time. It's a good kit, but it basically, if you scroll the numbers for every class that you ever taught, it's about 50-50. You have like 20 plates, 12 and 8, usually somewhere in that 8 to 12 region that are there. We talked about the reversion being so important. Again, it's a standalone question, the Ames test, using an auxotroph of salmonella that's histidine necessary, and it reverts back. And so you mix that with some liver enzymes, and voila, that's the test that's done on any consumable or medication that's there. And they go through all this. So, and obviously point mutations are a little easier to deal with and less likely to be problematic than, basically deletion or insertion sequences where you're going to inherently alter the downstream chain that's there. And I think most people know that. And obviously different kinds, the point mutation versus the silent mutation. So the point mutation, you're typically silent, missense in a regular, amino acid incorporated versus nonsense premature chain. Okay. So that's all. Standard stuff. There they are. Hello. A knockout mutation. Yeah. Aerobic environments. We know that. Frame shift mutations. Transposons. We mentioned that. Love the corn. I don't think they get into this with agents that induce it in general. What are the take-home points? I mean, any of these, and many of these were some of the original chemotherapy agents that were used like that. I mean, basically, this 5-bromo-uracil was first used. It was called 5-fluorouracil, so they halogenated. It was great. This is when we were kids, learned about it 45 years. Someone would say, oh, we're going to start Mrs. Jones on 5-FU. I love that. What did you say? 5-fluorouracil, of course. I'm just saying. I don't. Ultraviolet light, thymine dimers. Okay, which is non-ionizing versus ionizing radiation. Breaks in DNA, typically to reactive oxygen species. All that good stuff. And they go through all these. If you want to look through this PowerPoint, it's on there. And again, I do not think that that's something. There's, to me, these are the analog agents. And it's really, the reason I think there are questions that can come about that is we use base analogs as antivirals, and we use base analogs a lot as cancer chemotherapy agents. So I could see graph questions and basically sequence questions that come from it. Not so much about intercalating. There's the transposition, thymine dimers, blah, blah, blah. And again, and so you can see all these types of things, the genetic aspects that are there with the breast cancer genes that are part of it, etc. Here's the DNA polymerase proofreading, mismatch repair. I don't know how much they're going to do on light and dark repair or SOS repair. I mean, it might be worthwhile giving a glance to those items. I think we have in our eukaryotic cells a lot more mechanisms. That's why ultraviolet light, just to be clear, is very ineffective when it comes to, it's fine for retarding bacterial growth. So they use it a lot in the food. So you can lay stuff out under ultraviolet lights that basically will retard bacterial growth, but killing bacteria it's not. Once it's grown, it's not going to make colonies disappear. It'll stop colonies from growing. And we do a lab experiment. I don't know if you did it or not. I mean, it's fun because you do it with M-SMEG and Staph aureus and B sub. So you get, a mycolic acid capsule. You get a relatively tame bacteria like Staph aureus, and you get an endospore form. So you can look at those different. So it's kind of neat to see what it does, but it's pretty effective as long as it's not going. These are lab stuff, and I'm not good with that. The Ames test on the auxotroph salmonella, etc. And that brings us to really what I wanted to cover, and that, other than transpose, is to know these. Okay, transformation, the classic experiments, right? What was the original one? Griffith's experiment in the 20s. Thank you very much. They just didn't know what was doing it. And I'm sure, Dr. Hilbert gave a chapter and verse about that until they did, what was that, the Hardy and Chase, where they're the ones that were able to radioactively label the protein versus the nucleotide. So they could tell it was DNA and not a protein that was doing it. Classic. Transformation, important. Transformation and transduction, no contact that was there. This is order of frequency that it occurs, naturally. Transformation, the transference of naked DNA, very random, but it occurs. Transduction, a little more commonly. Again, I think much more important vis-a-vis lysogenic conversion. And then lastly, it's more about the F plasmid, I think, than the HFR cells. The HFR cells are the ones that are for genetic engineering. That would be the context of that question, because theoretically you can repeat it. They use recombinant, and it's funny. Did you know how long that's been around? Just anecdotally. I was at Seton Hall, and they asked us to do a paper, and I ended up with getting a topic of recombinant DNA. I asked our biochem guy, who was a priest, for a reference, and this was like 1971. So I mean, those recombinant strains have been around for a long time. He goes, "Oh, there's an article," and it's the old story. If they give you one reference, then you go back into that article and get all the rest of the references. Go to the library; they're all on microfilm. Print them out, take it home, write a paper. Oh God, the good old days. Now, the big one, plasmid transfer. A population of Staph aureus can become MRSA overnight because of F plasmid transfer. F plasmid minimum requirements has the ability to make the F pilus, and the ability to accomplish the transfer. It's a one-shot deal because once, and it's all about when we do it for genetic engineering, knowing where the origin of really the origin of when it splits apart and begins, and you can genetically adhere to that so the genetic material can get transferred. More than one plasmid can be transferred at one time, and and every time you accomplish the transfer, the new cell becomes F positive as it replicates F positive, and that's why you can do it overnight. So I'm sure you're all aware of all of that. So origin of replication becomes important, where you can find it. The idea of homologous recombination, that it magically inserts itself kind of where it came from, replaces the other one, which is very interesting. Probably has something to do with, I don't know, does it have something to do with restriction enzymes? I don't know. There's the experiment itself that you're obviously familiar with. Transduction, again, there's a lot of resources to study this. I'm never good on maybe I do not know really the distinction between generalized and specialized. Do they cover specialized? I would have to look. I'll make a mental note and look that up when I'm watching one of the soccer games. United in City at 7:30 in the morning. I'll be transfixed. And then there's the conjugation. And again, historically, they thought they didn't understand the concept between sexual and non-sexual. Why is this not reproduction? Because you're just changing it. It's not a form of reproduction. That's there. So that's where they began to understand the F biology. You always see that. And I think that's basically everything.
Is that why it was called sex pili?
Yes.
Okay.
Because it looked like it was longer than a fimbriae and shorter than a flagella. So originally they were pili. Pili typically are a little bit longer than typical fimbriae microscopically. So they called it, because originally they thought, yeah, they put it under the microscope. Oh, don't look. Just saying. I think that's funny. And interesting, there are even some eukaryotes. That's where the yeasts come in when they do genetic engineering and all the other stuff that are about it, all these plasma encoded traits. Okay, R plasmid means it has resistance. So an F plasmid can be an R plasmid as well, but an R plasmid would have to have the F material to become an F plasmid. So it's a little bit different. And then, with regard to how these are inserted and things like that, that to me is more cell and molex and genetics, and I'm not the one to teach you about that. Interesting. We think that this transposon is responsible for vancomycin resistance. And that's a real problem because vanco is the drug of last resort. And and I tell everybody, I think I told you, when all of a sudden they're telling you can't use the vanco, that's significant. We see vanco being used more like in nursing home populations, and you have to monitor it relatively closely. You have to do peaks and troughs and things like that. Who knew? Again, I'm not a plant guy. Pathogenicity islands, just areas genetically in that, and any of these genes that move in the mobile gene pool. Lastly, the big area, the review of gram positive and gram negative. And this is all that's all the microscope stuff. I mean, you can read that as well as I can talk to you. And the different staining and things like that. Could there be a standalone question based on that? Possibly, basic stains, because they're basic chemically, okay, and positively charged, and effectively their adherence is typically to things that are negatively charged and acidic in nature as components of the cell wall. That's where that all comes from. Acidic or the reverse or negative stains, they repel. So, my analogy for that, those background staining is what you would call it, using things like nigrosin or India ink or Congo red, one of my favorites. And it's like a light bright. I'm thinking granddaughter. I'm thinking light bright. And so, yeah, I know the mechanics of the gram stain. What the sequence? That's that, all those big major stains. So whether you know the mechanics, you know the crystal violet and the saffron, and you should know for the acid fast stain, okay, the malachite green and the saffron, and because differential means they're two opposing colors, and even the carbofuchsin. The old days we used to call it Castellani's paint that were there, and the methylene blue for the acid fast stain. So I mean, just bang, bang, bang. I would know those big guys. Methylene blue, you also use. It's kind of it's sort of a random absorption of stain, and it's very useful for showing those metachromatic granules that are associated with corynebacteria, which is kind of interesting. You saw a lot of that. Corynebacteria was sort of the foundation bacteria for diphtheria, and we don't see a lot of it today. That's another interesting disease. There it is, back capsule stains, background stains, endospore stains, special stains. And the flagella, obviously, the whole idea of flagella is mostly for identification. Okay, I mean, you get some idea if they move. Things that have multipolar flagella move a lot. The one we use in our lab here, Proteus vulgaris, Proteus mirabilis, those guys move around quite a bit. That are there. And then, of course, the naming convention. Coxeye rods and spiral forms, that's it. The spiral forms have the subcategories, the spirilla and the spirochetes that are there. They all can be gram positive and gram negative. They move differently. Remember, the spiral forms, the flagella, are inside. It's sort of like, it's like, it's like in the old day, in surgery, we used to use a thing called a giggly saw. So when you were doing an amputation, you didn't have some people used a regular saw. They used saw blade teeth, and you hooked them onto two like hemostats, and you wrapped them around the bone and went back and forth. That's kind of like that spiral action that you see, because those are, and you'll see them where the flagella are sort of within the bacteria rather than protruding from it.
So that amputations then, or
pardon me?
So whenever you're saying like with that law off topic, but like so would you do amputations like a lot, or is it kind of like a car accident type?
We had a lot of older people with vascular problems.
Oh, okay.
And really, in those days, the vascular surgery, we were dealing with an indigent population. Half the people or more had diabetes, late stage. They drank, they smoked. It's 1977.
Yeah.
It's like it's just everybody. Well, I chain smoked. I loved it. You did it again. My mother and I both loved to smoke. It was awful. I told you. I think I told you, but in my class, in my house, people go. You do this. You either a deck of cards or an ashtray. It's a steady joke. God bless you, Ruth and Sid. Okay, so I don't think they're going to get too much into these, but basic. I mean, if you should know. Yeah, Treponema pallidum is the classic spirochete. Lyme disease, Borrelia burgdorferi, classic. What's interesting about those from a clinical standpoint is that your response to them, unlike most bacteria produce toxins, those bacteria, it's immunological. Okay, so you get, and that's the distinction between direct and indirect effects. In bacteria, direct effects are toxins. Staphylococcus, streptococcus, they make toxins. Botulism toxin, tetanus toxin, versus the indirect effects, which means your immune system. So it doesn't preclude some of those toxins having immunological activity, but by and large, not all of them do. So I mean, you kind of get into both of those. So a lot of times the toxin's bad, and plus it adds to there's an immunological response to it, and you can get something that fulminates. Maybe that would be like gas gangrene, which we don't see a lot of now because we don't have a lot of these war injuries. They were so common. Again, that's back to me being a World War II buff. where there were so many amputations. So all of that. Vibrio for cholera is the most noted. Not a whole lot of pathology with spirillum. These pleomorphs don't have a very stable cell wall. And that's where mycoplasma comes in. And mycoplasma is one of the most common kinds of pneumonia that's bacterial, walking pneumonia, very common in college populations. And they look differently because without that defined cell wall, then they just sort of shape themselves to the environmental pressures. So they have sterols, okay, that make that. Sterols are interesting microbiologically because when you get into eukaryotic pathogens, that we target sterols. So for fungi, the way that we treat fungi, whether it's topical or internally by injection or orally, is they're all target ergosterol, which is the sterol. Our cells are cholesterol. Okay, but fungi is ergosterol, and that's a very useful target. That's the good news. The bad news about treating eukaryotic infections, whether they're, for the most part, whether they're fungi, whether there's worms, whether they are protozoans, okay, is that unfortunately those cells are similar to ours, so they're much more likely to have side effects. And some therapy isn't as bad, and some therapy is very bad. That's why for years there was very, very little. The antifungals internally were very, very harsh. There are better ones now that are available. So you could do, I mean, people can take those for athlete's foot and for fungal nails or for deep fungal diseases like San Joaquin Valley. Now they call it valley fever. It used to be called San Joaquin Valley fever. Most of those are encapsulated fungi. And again, those are the kinds of illnesses, depends on where you live. Okay. So, I mean, so if you go on into a medical field and go into the Southwest, you've got to know about the diseases that are there. If you're in the deep South, you have to know about the diseases that are there. So there's a certain regional aspect to those things. Like, so when we talked about, we happen to be talking about like deep fungal diseases, and when you're looking at those guys, those are sterols are what are holding that bacteria in place. It doesn't have it has peptidoglycan, it's just not a well-defined cell wall. And we use ergosterol and ergosterol targets for fungi. But if you, like I said, my brother-in-law, my wife's brother lives they've lived in Arizona for forever, Tucson. I mean, it's not, you talk about valley fever, their dog gets it. Because there's a whole different thing. You worry about things like undulating fever, which comes from rat droppings. Okay. You have to be you're sort of on top of some of those kinds of diseases that are there. Moving on. And Staphylo and Strepto are the ones to be familiar with. And it's all about how they replicate and how they adhere randomly or in planes, etc. And now we're getting to the fun part. We know about that. Again, flagella, pili, and the shorter ones are called fimbriae. Not a lot of moving parts. You know, supercoiling, that's the beauty of the nucleus. It's ready to go. It's haploid. It's double-stranded DNA in a circle that's supercoiled, ready to go, no pairs. That's the beauty of it. The good news for replication, the bad news for being damaged in mutations that are there. Ribosomes. So the nucleus, as you can see stain-wise, predominates. They're there, and they go through all this. I just want to get into that. Membrane, the same, nothing unusual about that vis-a-vis our membranes. The archaea, the difference is they don't have peptidoglycan. There's some differences within that I don't think are terribly important, but the environmental nature that they exist in extreme environments. As an aside, their DNA sequences are more similar to ours than bacteria. Just curious. Yeah, we did the whole. I'm not going to go over the diffusion and non-diffusion and all that kind of stuff. Active transport. I mean, you can scroll through these. Bacteria, they have very little to me, since most of them ferment. There's not a whole lot of energy to do active transport activities other than are necessary. So they fermentation is sort of is the default for a lot of bacteria because oftentimes they exist in conditions that aren't conducive. Every so often you'll get ones, and we get some like Pseudomonas, which is a real problem in a hospital. Okay, is an oddball aerobic gram-negative. Just for whatever reason, it's flourished because it came in plants and on the bottom of your shoes and things like that are there. So there's a fair amount of stuff that goes. So they go over all that in length. Gram-positive v gram-negative. Super important, I think. Core questions for any knowledge of micro on an Mcat sense. Not just the color of what it is or typical genera that do that. The peptidoglycan. Okay, with the exception of those oddball things like the mycoplasma, they all have, we either fall into gram-positive or gram-negative, with rare exceptions. Gram-positive, thick layer, teichoic acids, which do serve to adhere this relatively thick shell to the plasma membrane. To me, I love the term when they use cell envelope because they're a unit. If you break the wall, the envelope breaks down. If you damage the membrane, the envelope breaks down. They function as a unit. So it's pretty significant to me. That's why cell wall antibiotics work so well. They don't have, this is a typical plasma membrane, the gram-negative. It's all about that outer membrane and the outer layer of the outer membrane, that LPS, that lipopolysaccharide layer. That's unique. And again, unique mean ain't nowhere else. Peptidoglycan is unique to bacteria. The LPS layer is unique to gram-negatives. And its component, the lipid A, the lipid portion of that lipid is a toxin to us. And it's and that's by definition an endotoxin, meaning all bacteria can make exotoxins, whether they're gram-positive or gram-negative. If they use the minute you see endotoxin, you know it's a gram-negative. If they say, gram-negative sepsis, you know it's an endotoxin. That's an immunological reaction to that endotoxin. Gram-negative sepsis sucks. Ruptured appendix, that's what you're looking at. Rupture from cancer, whether it's ovarian, bladder, gastrointestinal, all of those. Perforated ulcer, that's all going to, that's an immediately life-threatening situation. Okay. So inherently it's porous, so you don't need porin proteins for water to move in and out. Okay. There's no outer membrane, you don't have to worry about it. And yes, the watchword penicillin, cell wall type antibiotics. So your big players here to know about penicillins and cephalosporins. Those are the big players in the cell wall. Anyway, the most versatile of these by far are cephalosporins because they have more activity against enzymes. They have beta-lactamase-based enzymes. Okay, they have not so much penicillinases, but beta-lactamases, which are subtly different. So non-staphylococcal organisms and some staphylococcal organisms don't make penicillinase, they make beta-lactamase. And so cephalosporins, they're much better on gram-negatives. They're broader in spectrum. Nowadays, they're relatively, and we've tinkered with them enough that we're starting to use them more for MRSA that are there. They are, here's the nugget. If you look at all the prescriptions written, let's say in the US in a year, around half of them are for cephalosporins. So in the exception of people who are allergic to penicillin, there's about a 10% cross-reactivity with cephalosporins. It's when in doubt, Keflex. It's like the go-to thing that you're going to, Cephalex. Right now, it's cheap as chips. Years ago, when I first started in practice, we had to worry about that. People didn't have insurance plans. There was nothing. They didn't have, even the elderly. I can tell you how many elderly people said, yeah, I had to find them a prescription they could afford. It was them eating or getting their pills. Just say, here, a very 30 to 40 layers here, thin here, comparatively. sandwiched within the membrane with a fluid, very interesting, called periplasm. A lot of stuff goes on in the periplasm. How it beats, how it develops resistance, efflux pumps. Stuff comes in and pumps them back out, antibiotics. It's very neat in the periplasm. Okay, pores or porin proteins allow transiting of water. NAMs and NAGs, remember, you won't have to know the names, but they're alternating glycans, nitrogenated sugar-like substances. And the only time you see the tetrapeptide chains and the only time you see the interbridges are in gram-positive. It's the bricks and the mortar analogy that I use. The bricks are the peptidoglycans, polymer. The mortar that holds it, says you have a brick wall, no mortar, you can break through it. The mortar that holds it together, to me, that's the tetrapeptide chains and the peptide interbridges. Voila. And again, secured to some extent by the teichoic acids. They're there. Love the illustration. Ooh. Okay. The difference is that outer layer of the outer membrane. Here's the one or two layers, relatively thin, really doesn't use the interbridges that are there. And the interesting stuff that goes on here, normal, good old phospholipid bilayer, good to its mother, always calls her on her birthday. I hate everybody. I don't like anybody. That's why I like it so much when the trio comes in.
Rattle the cage.
They rattle you. You're awful. Take my students, please. That's the old joke. Henny Youngman. Is the king of the one-liners. Take my wife, please. And he has some really good dirty jokes.
You should watch one of their specials sometime.
He's dead.
But that doesn't mean he didn't make a special. He's like,
Oh, I watch them. They're on YouTube, all these things.
We should take a day off
and do YouTube.
Mental health day. We could watch on
my mental health is beyond repair. The outer portion. This is the lipopolysaccharide layer because it has that lipid A, the unique endotoxin, and the polysaccharide has to do with this. They used to call it the O-specific polysaccharide. Now they call it the O-antigen. And in this case, again, protruding sugars for identification. And so we are able to type it with that. And again, on the flagella, there are flagellar antigens as well. So when you see O and H, the very famous E. coli, O157H7, that's how they identify it. So that's the relevance and the importance of that. There's about 6 kinds of E. coli that have those really significant, potentially life-threatening toxins, and periplasm, transport systems, penicillin, lysozyme also attacks peptidoglycan. Preventatively, there's the gram stain, slime layers, importance too. If it's very, very well organized, you typically get the capsule, and the capsule, very significant in respiratory diseases. Tuberculosis, Klebsiella pneumonia, big capsule, streptococcal pneumonia, big capsule. The one strains that have that, like in the Griffith experiment, the Hardy and Chase experiments, those are the ones that lead to bacteria. And bacterial pneumonia is the major killer post-viral infection, influenza, COVID, etc. So there is a certain degree of importance. The slime layers are very important in their utilization. Rather than being thick, they're relatively flimsy and sticky. They adhere to one another. They create those biofilms, and so much goes on with them. In the old days, we had a bathtub or a toilet bowl. It was slime that was on rocks and things when we were out hiking, and with the stream and stuff like that. I remember taking some pretty good falls. We were like down at the Yacht, whitewater rafting stuff. I wouldn't go to rafting. My aforementioned brother-in-law, my wife calls him Psycho Bob. He's the same age I am. He rides his bike like 50 miles a day. He's got no waistline. It's one of those. Remember, this guy's waist the same as he did in high school.
I think every town has one of those.
Oh,
it's my buddy's grandfather. His name's Mick, local legend.
When he stays at my mother-in-law's, he zooms down in North Park. He's flying around on his bike, crazy. So the flagella, again, run, tumble, run to remember. The flagella arrangements are for identification. Single polar is probably the most common. The little tufts are lophotrichus. If you have two of them, they're amphitrichus. And the peritrichus generally is the swarming. And that's what we use for identification. This is pretty straightforward. You have the base area, the hook, and the filament itself. Totally different extruding than our internal flagella. Okay, those project out. They're not covered in membrane. Ours are made of microtubules. These are made of flagella. Pili and fimbria are made of pilin. They protrude. They're not analogous to our cilia, which are made of, again, of microtubules. Well, that 9+2. That's so important to remember. Good old run, tumble, run. Mostly chemotaxis, some aerotaxis, photosynthetic bacteria. Okay, typically have little air kinds of, they're like vesicles, but for buoyancy. So they float. Exotic bacteria have those magnetites, so they're attracted by magnetic. Various bacteria are thermally attracted. This is when you see the temperature levels. And remember all those. Most of the ones that impact us are mesophiles that cause disease. There's only about 2,000 strains we identified are really truly disease causers, 2,000 species. And they're typically their life cycle is greatest as mesophiles at around room temperature. So they basically are in that realm of maybe like 40 to 75 degrees Celsius. Okay. In that, we're in that area. Okay. It's when they get higher than that, you have to understand that, I beg your pardon. I flip-flop that. That's the thermophiles, the ones that are like hot tubs and hot springs. So they're like from 20 to 45 Celsius. So they're around 37 degrees Celsius is their peak. Remember that curve. It's not a bell curve. Bell curve is how they respond to pH. Mostly neutrophils, not neutrophils like white blood cells, but they're in that pH 5 to pH 8, 6 to 8 range, around pH 7. But the thermal, and we use this all the time, it's why heat is so effective. Basically, on the cold side, it comes up like a bell curve, but the optimal temperature is very close to the death temperature, like us, and then boom, falling off a cliff. That's the classic. That's why temperature is so effective. Freezing, helpful to retard growth, not helpful for killing. Temperature, cheap, and all that type of stuff. So they are adapted to temperature, and certainly photosynthetic bacteria deal a lot with light as well. The pili, the fimbria are mostly attachment. Plasmids, for the plasmid movement. Most of plasmids non-essential but useful information, can come and go in a population as needed, typically more than one. 70 versus 80S, so terribly important. Okay. Again, that's the endosymbiotic theory. And chlorophylls and mitochondria being, probably prokaryotic organisms that became incorporated and adapted. Their DNA is single circular. Granules for identification. Endospores. As far as we know, gram-positives don't do them. Gram-negatives, I beg your pardon, don't do them. They're only in gram-positives. And that's part of the main classification for gram-positives. First step, endospore or non-endospore form. Basically two. There's some exotic bacteria that have a kind of sporulation. There's soil bacteria. They're where a lot of antibiotics come from, but antibiotics also come from bacillus. So the one we use in the lab, B sub, that makes bacitracin, which is originally part of triple antibiotic, which was developed where? Pittsburgh. Burroughs Wellcome Company. And now they've all kind of swallowed each other. Little bits of history. Just saying. So, I mean, I'm from New Jersey. We had so many chemical plants and everything. I mean, you could walk across the Passaic River. And it wasn't because you were religious. You could walk. It was solid. Bacillus and Clostridia. Difference, Bacillus aerobic, easy to cultivate. Clostridia, very significant medically, with tetanus and botulism, but it invades dead tissue, okay, or an anaerobic environment. Clostridium perfringens and histolyticum for gas gangrene, and very famously C. diff, which is because of the anaerobic environment of your colon, and that can flourish with antibiotics. It's sexually indestructible other than with extreme conditions, most commonly sterilization, the removal of all of those. So it's not reproduction. It's just a transition from its vegetative stage, which means it's growing. Remember, we thought they were plants. So when a bacteria is vegetating, it's growing, it's happy. I think I'll vegetate today. Oh, lucky you. Versus endosporing, where you're kind of going to a state of what we used to call suspended animation. And so they're everywhere. Go through that, all that. So you can review that. There's a really good animation. If you can't access it, give me a holler. Basically about the forespore, which becomes the endospore, and this deteriorating. So the replicated DNA deteriorates, and then peptidoglycan is always laid down in the middle. So effectively, they're gram positives. And name number, da da da. Yeah. And some of the distinctions between eukarya are important. Things that they do and don't have, I think, are important as well. Obviously, the distinction between animal cells and plant cells, cell walls. They don't have the thing that makes the microtubules, the centrosome. They don't have those big gas vesicles that are there. I mean, those are some of the distinctions. Obviously, the chlorophyll. I don't know. Again, I'm not really good on the plant aspects of it. But that's why you have Dr. Bilwick. We did all that, did all that, did all that, did all that, did all that. 9 plus 2. Endosymbiotic. Okay. Circular DNA, ribosomes. Same thing with again, endosymbiotic theory, something to review. Same deal here. Okay. Cell wall and replication as partners because indispensable to one another, etc. DNA, ribosomal RNA, 70S, double membrane around both, binary fission to replicate. Okay. Mitochondrial DNA sequences resemble those of, and again, the rickettsia years ago, rickettsial diseases, most notably Rocky Mountain spotted fever and typhus, were thought to, when I learned about them in 1974, they were thought to be, we didn't know they were bacteria. We didn't have the molecular biology. They were these are rickettsial diseases, and they're typically carried, this is how we learned it, by lice and ticks. That's where you learn the name. And now we see the relevance of them. Okay, I'm done. 10:50. That wasn't bad. That means on Wednesday, you know who you're going to have? Really? Mike, if you want, it's up to you. If you want to drop in on the other, I don't know if you're around at 9:00.
Yeah, I just saw the email this morning, so I was
Yeah, I mean,
already behind the ball.
The issue, everybody, is the registrar goes nuts with registration and stuff like that. So check and see what they're talking about if you want to drop in. Make it informal.
Yeah, that's I think what I'm start doing.
Yeah, informal. Okay. So I mean that's we're trying to get stuff to get you prepared just like we get everybody else, try to get everybody else prepared. So I think it's important. Nevertheless, that's all it is. Goodbye. Have fun. No, you're goodbye. I'm stuck with them for another hour. Then the dog is getting groomed. Puppy haircut.
Oh, yeah. Where do you go with it? Are you in the camp of like getting them shaved so they're cooler for the summer?
No. I just he needs to be. It gets out of control. The
Yeah.
doodles out of
Oh,
control.
yeah.
He's a doodle.
Is our golden retriever